The genetic information necessary to build proteins is contained within an organism's genome. To create a protein, genes are first transcribed to create messenger RNA. The mRNA is then read by a large structure called the ribosome, which links amino acids together to form the protein encoded by the mRNA. The genetic information necessary to build proteins is contained within an organism's genome. To create a protein, genes are first transcribed to create messenger RNA. The mRNA is then read by a large structure called the ribosome, which links amino acids together to form the protein encoded by the mRNA. The ribosome cannot accomplish this feat unaided, however. Besides the mRNA and a ribosome, translation also requires amino acid carriers called transfer RNAs and a large number of initiation, elongation, and release factors. Finally, ribosomes cannot be reused in a cell without the action of a recycling factor. The ribosome is a large macromolecular structure that performs protein synthesis in cells. The ribosome links together the appropriate amino acids to form the polypeptide chain encoded by the gene. The ribosome consists of two subunits, the small subunit and the large subunit, each consisting of both ribosomal RNA and protein. In contrast with many enzymes you have studied, over half of the ribosome is composed of RNA, with protein making up a smaller portion of the mass of this enzyme. Ribosomes are similar in structure in prokaryotes and eukaryotes, although the overall size of the ribosome is smaller in prokaryotes. Ribosomal subunits are named by their Svedberg coefficients, a measure of their sedimentation rate when subjected to centrifugal force. Ribosomal subunits assemble into a full ribosome on the mRNA, disassemble when the protein has been synthesized, and assemble once again to start another round of protein synthesis. This process of repeated assembly and disassembly of ribosomal subunits is known as the ribosome cycle. The ribosome reads the mRNA 3 bases at a time. Note that, while spaces have been added to aid in visualization, these triplet codons are immediately adjacent to one another on the mRNA. The triplet codons are matched within the ribosome to specialized RNA molecules that carry amino acids. These amino acid carrying RNAs are called transfer RNAs, or tRNAs, for they are adapter molecules that transfer genetic information to amino acid sequence. tRNAs contain anticodons that are complementary to, and thus bind, the codons on the mRNA. The ribosome is uniquely designed to bring mRNA and tRNAs together. The mRNA is threaded through the small subunit, while the large and small subunits of the ribosome fit together to make tRNA binding pockets. The ribosome contains 3 binding sites for holding tRNA. The A site accepts tRNA molecules charged with single amino acids. The P site contains the tRNA linked to the growing peptide chain, and the E or exit site is occupied by a tRNA molecule about to be released. The growing protein exits the ribosome through a tunnel in the large subunit. For translation to begin, the ribosome needs to be seated properly over the start codon of the mRNA, with a charged tRNA in the P site. This process of initiation, which differs significantly in prokaryotes and eukaryotes, is aided by a number of translation initiation factors. First, we will describe prokaryotic translation initiation, which requires 3 initiation factors, IF1, IF2, and IF3. Each binds the 30S small ribosomal subunit. Initiation factor 3 binds the 30S small subunit and prevents association with the 50S large subunit. This keeps the 30S small subunit open for mRNA binding. Next, initiation factor 1 nears the A site, and initiation factor 2 binds both the 30S subunit and IF1 in association with a GTP molecule. Next, both the mRNA and initiator tRNA can assemble onto the complex. Initiation factor 1 blocks tRNAs from improperly entering the A site, and IF2 brings the initiator tRNA charged with F-methionine to the P site. The ribosome binds to a sequence on the mRNA, called the Shine-Dalgarno sequence, through a complementary sequence on the 16S RNA of the small subunit. The messenger and transfer RNAs can bind the 30S subunit in either order. The small subunit of the ribosome, aligned with the mRNA start codon, is called the 30S initiation complex. The binding of the charged FMET tRNA to the small subunit initiates a sequence of events to form the 70S initiation complex. Once the FMET tRNA has bound the small subunit, IF3 falls off and is replaced by the large subunit, which completes the ribosome. Large subunit binding stimulates the GTPase activity of IF2. Specifically, the factor binding center on the large subunit stimulates IF2's GTPase activity. Because the GDP-IF2 complex has a low affinity for the ribosome, IF2 and GDP, and then IF1, are all released. This is the 70S initiation complex. Protein synthesis can now begin. In eukaryotic translation, initiation is more complex and uses at least 8 initiation factors compared to 3 in prokaryotes. To begin, the small ribosomal subunit binds to the 5' end of the mRNA, called a 5' cap, and then scans along the mRNA to find the start codon. After an initiation complex is formed, elongation occurs. During elongation, the ribosome synthesizes proteins by linking together amino acids. 3 steps are needed to add each amino acid. A charged tRNA is placed in the A site of the ribosome. A peptide bond is formed, and the tRNA with the growing protein chain is moved from the A site to the P site. In prokaryotes, this requires 2 translation elongation factors, EFTU and EFG. A third elongation factor, EFTS, is needed to recycle EFTU. Elongation is very similar in eukaryotes, and only the prokaryotic process will be described here. When the triplet anticodon of the correct tRNA is base paired with the mRNA in the ribosome A site, the base pairing is strengthened by additional hydrogen bonding with the ribosome. Incorrectly bound tRNAs do not base pair as strongly with the ribosome and are released before they can be added to the growing polypeptide chain. Correctly base paired tRNAs have additional hydrogen bonds that help the tRNA remain in the A site during accommodation, the process whereby the tRNA rotates into position for peptide bond formation. First, a charged tRNA is bound by EFTU and is brought to the ribosome A site as an EFTU GTP aminoacyl tRNA complex. The tRNA anticodon binds the mRNA codon, and EFTU hydrolyzes its GTP, and the resulting EFTU GDP is released from the tRNA. Next, a peptide bond is formed between the amino acids. This reaction transfers the amino acid from the tRNA in the P site onto the aminoacyl tRNA in the A site. To prepare for addition of the next amino acid, the ribosome undergoes translocation, a process in which the ribosome moves along the mRNA to the next codon. Translocation is facilitated by elongation factor EFG. The tRNA with the growing amino acid chain attached moves from the A site into the P site, and the spent tRNA in the P site moves into the E site. Note that elongation factors EFTU and EFG both end up as complexes with GDP after one round of elongation. Both factors must exchange their GDP for GTP before they can be used again. For EFTU, this exchange is facilitated by elongation factor EFTS. For EFG, the GDP simply falls off and is quickly replaced by a GTP. The process continues. At each codon, the corresponding aminoacyl tRNA binds in the A site. The growing peptide chain attaches to the amino acid on the tRNA in the A site, and the ribosome translocates, moving the growing protein chain and tRNA into the P site, and freeing up the A site for another round of amino acid addition. Termination of translation is engaged when a stop codon on the mRNA enters the A site of the ribosome and is performed by release factors that recognize the stop codon. The mechanism of termination is similar in prokaryotes and eukaryotes and involves two kinds of release factors called class I release factors and class II release factors. Class I release factors enter the ribosome A site and bind the stop codon using a peptide anticodon. Prokaryotes have two class I release factors, RF1 and RF2. Eukaryotes have one class I release factor, ERF1. The class II release factor, in complex with GDP, also binds the large subunit. In prokaryotes, RF3 is the class II release factor. In eukaryotes, ERF3 is the class II release factor. After the class I and class II release factors are both bound, a glycine-glycine-glutamine, or GGQ motif, on the class I release factor triggers hydrolysis of the protein from the tRNA in the P site. Release of the protein from the ribosome triggers the recycling of the release factors. First, GTP is exchanged for GDP on the class II release factor. This causes the class II release factor to push the class I release factor off the ribosome. The release of the class I release factor allows RF3 to interact with the factor binding center, which stimulates RF3's GTPase activity. GTP is then hydrolyzed on the class II release factor. The class II release factor GDP complex then releases from the used ribosome. At this point, the ribosome still contains two deacetylated tRNAs and is also still bound to the mRNA. The process of cleaning out the ribosome and separating the subunits is called ribosome recycling. Although not as well understood in eukaryotes, in prokaryotes this process involves a ribosome recycling factor plus initiation factor IF3 and elongation factor EFG. The ribosome recycling factor, RRF, binds to the A site of the used ribosome and recruits EFG to the complex. The RRF acts like an A site tRNA during the translocation process. EFG can thus push on the RRF in the same way as it would on a tRNA. This moves the RRF into the P site and releases the two spent tRNAs. Next, both the RRF and EFG uncouple from the ribosome. The mRNA is released, possibly with the aid of IF3. IF3 binds the small subunit to prepare it for another round of protein synthesis. Many antibiotics work by selectively targeting the bacterial ribosome, blocking translation. Because these medicines are unable to bind eukaryotic ribosomes, antibiotics